Good morning, everyone. First, I'd like to thank the organizers, Vani and Andrew, for inviting me to participate in the symposium. I'm honored to be a part of today's speaker lineup. So the work that I'm going to be presenting today is some recent work led by my brilliant PhD student, Parker Singleton, who's in the computational biology department at Cornell. And a full description of the work can actually be found as a preprint on bioRxiv. So understanding and being able to objectively measure the mechanisms of psychedelics is really important if we aim for their use in a therapeutic context in which it's increasingly being used. A decade of neuroimaging studies has provided some novel insights regarding the psychedelic action in the brain. And one relatively consistent finding is that brain activity demonstrates an increase in entropy under the effects of psychedelics. And this seems to be related at a regional level to both the amount of white matter connectivity that we see a region has, as well as its 5-HT2A serotonin receptor density. This is where psychedelics have their most robust and agonistic effects. One model that was proposed recently in 2019 by Carhart, Harris, and Friston, it's called the RElaxed Beliefs Under Psychedelics hypothesis, and it was created in part to explain these findings. It actually views the brain as a Bayesian model or prediction engine, whereby perception and belief are shaped by both prior knowledge that you have about the system and the environment, as well as the incoming sensory information. Within REBUS, the psychedelics are postulated to translate into a decrease of weight on those prior beliefs, which then has an enabling implication for bottom-up information flow. So it's theorized that the observed increase in entropy of brain activity under psychedelics is actually reflective of reduced energetic demands for the brain to navigate its dynamic landscape. And this work, as proposing REBUS and others finding increased entropy of brain activity patterns, led us to our current hypothesis. So our hypothesis that we're testing with this work is that perhaps psychedelics' observed effects of increased entropy in brain activity actually arises from a flattening of the brain's energetic landscape. In this scenario, we imagine brain activity is represented by the coordinates of a hiker within a landscape. The shape of the landscape and how steep the slopes are will determine how much energy is required to move from one point in the landscape to another. We actually hypothesize that under psychedelics, the brain's energetic landscape may flatten, resulting in more easy and frequent state transitions of brain activity and thus increased entropy. So the approach that we took was one that was very eloquently proposed in Kornblath et al. In 2020 that uses network control theory to characterize brain states and quantify the trajectories of brain activity through the state space. Their paper, interestingly, linked the amount of energy required to move in the brain state space to measures of working memory performance in healthy controlled population. But most importantly, this approach actually allows us precise measurement of the brain's energetic landscape and a testing of our hypothesis that the landscape flattens under psychedelics. So in network control theory, the brain is considered to be a linear system in which activity that's represented by the vector x at time t flows both passively through the network via a diffusion process on the structural connectome, here represented by the matrix A, and actively through the effect of external inputs represented here by the product of the control matrix B and the external driving input U of t that is temporally dependent. After computing the controllability Gramian, defined here as W, we can solve for the amount of energy, or the E min, that represents the minimum amount of energy that's actually needed to change the course of the natural diffusion process and complete that state transition from our initial state x naught to our final state xt. So let's get to the data and the experimental design. So this data was provided to us by our collaborators and collected in 2016. This first study was studying the effects of LSD. So these were 20 healthy volunteers who underwent 2 MRI scanning sessions at least 2 weeks apart. On one day, the participants were given IV placebo, and on the other day, they received LSD also intravenously. It was infused over 2 minutes, and right after infusion, the subjects had a brief acclimation period in a mock MRI scanner. And then 115 minutes after infusion, they went in for the full resting state scan. They had 3 7-minute and 20-second eyes closed resting state scans. The first and third were under rest, and the second one involved listening to music. Of the 20 individuals who participated in this study, 1 was excluded for anxiety and 4 for excessive motion. So we had 15 subjects left. The other study was a psilocybin experiment. And here we had 9 volunteers over 2 days. They were either given placebo or intravenous psilocybin and immediately scanned after administration of both substances with 5 minutes of resting state fMRI. So after collecting the data, we ran it through all of our processing pipelines, including strict control for motion, which was an important element to this study. After we had our clean data, we calculated the regional time series and concatenated all of the subjects' placebo and LSD or psilocybin scans into a big matrix and applied k-means clustering to this matrix to identify commonly recurring states of coactivation. And here we show the 4 states that we'd found. We tested for optimal number of clusters using elbow criteria and increase in explained variance. And we found 4 to be the optimal number of states. And when we looked at these states, we see that they are really 2 meta states that are composed of activity that is anti-correlated. So here we have meta state 1, which is defined by high amplitude and low amplitude activity in the somatomotor and default mode networks, and meta state 2, which is dominated more by frontal parietal activity, high and low frontal parietal activity. So once we have the brain states defined, we can actually then calculate metrics such as fractional occupancy, dwell time, appearance rate, and transition energies between states. So here, fractional occupancy is just the amount of time that a person spends in any given state. Dwell time is the amount of time that is spent in that state once the brain arrives at that state. Appearance rate is the number of times per minute that state is traveled to. And transition energy is, as defined before, the amount of energy required to inject into a system to complete the state transition from one state to another. So first, we see a shift in temporal dynamics under psychedelics. You can see here that there's an increase in fractional occupancy of metastate 1 under LSD compared to the psilocybin case, and a decrease of fractional occupancy in metastate 2 under LSD compared to placebo. Again, this metastate 1 was somatomotor and default mode driven, while metastate 2 was more frontal parietal or a higher order activity. And we see that the shift in fractional occupancy, at least in LSD, may be explained by changes in dwell time. So the amount of time that is the brain spends in a state once it gets to that state. We also see a similar shift in psilocybin case, except this wasn't statistically significant for fractional occupancy. And those sort of non-significant changes in fractional occupancy seem to be dominated mostly by changes in appearance rate. So to our main and primary hypothesis, we see that both LSD and psilocybin actually lower the state transition energies between all pairs of states. So it seems that across the board, there's a decrease in the amount of energy needed to complete the transitions between every state and every other state in both LSD and psilocybin compared to the placebo case. We actually see that across individuals, there is a correlation between the amount of energy reduction in a particular subject by LSD and the decrease in dwell time and the increase in appearance rate and the increase in entropy. So those subjects that had larger decreases in energy between the placebo and the LSD case also had a larger shift downward in dwell time and a larger shift upward in appearance rate and a larger increase of entropy as measured by the Lempels of complexity. So the next question we had was by what mechanism do these transition energies decrease? So in the network control theory results of transition energy calculations that I just showed you, our control vector was a uniform control vector, which means that each region in the brain had uniform control over the system. But we also know that the LSD and psilocybin act on a specific serotonin receptor, specifically the 5-HT2A receptor. So we decided to test to see if using the 5-HT2A receptor density maps could also result in a decrease of energy, transition energy in this network control theory framework. So again, here was the result using a uniform control vector input. And here's the result by using the placebo centroids and just shifting the control weights to be from uniform weights into the 5-HT2A spatial density receptor map. And we do see that when we use the control vector derived from the 5-HT2A density, we see a decrease in overall energetic demand for transitioning between brain states. We wanted to test that the distribution of these shifts in energetic demand was not because of the spatial distribution and the weights changing. So we did a spin permutation test where we randomly permuted the 5-HT2A density receptor map values and calculated that against the true distribution. And we do see a persistent decrease in energetic demand to transition between brain states. We also wanted to compare other serotonin receptor maps to the 5-HT2A map in its ability to flatten the energetic landscape. And we do see that compared to 4 other serotonin receptor spatial distribution maps, that the 5-HT2A receptor map does provide an optimal flattening of the energetic landscape. So this multimodal study actually linked receptor density or PET imaging of serotonin receptor maps and anatomical connectivity to whole brain changes in brain activity dynamics at rest under the acute effects of psychedelics. The shift that we see in brain state occupancy, namely this increased occupancy in somatomotor and default mode network states and less occupancy in frontal parietal states, was consistent across the LSD and psilocybin studies. This work provides evidence for the hypothesis that there is a flattening of the brain's dynamic landscape and that this flattening could result in easier and more frequent state transitions and an increase in brain activity entropy. The spatial distribution of the 5-HT2A receptor that we know is impacted by LSD and psilocybin seems poised to allow for an optimal flattening of the brain's energetic landscape compared to other serotonin receptors. The approach that we used here of network control theory and PET imaging could be used in the future to quantify or predict the effects of receptor-specific pharmacological compounds to changes in shifts in whole brain activity and dynamics. Finally, I'd like to point out that about 75% of people list their psychedelic experiences as the top 5 most meaningful life experiences that they've had. However, there can be bad trips that are emotionally negative or harmful to the psyche, and seemingly only occurring in 4% of chronic users. Some people have reported hallucinogen-induced persistent perception disorder or flashbacks. So we need to be careful while using these powerful neuromodulators. Particularly, there has been an increased interest of commercial and academic and public interest in the use of these substances for treating psychiatric disorders such as depression, substance use, eating disorders, or PTSD. So I believe that a detailed understanding of the mechanism and the long-term impact on the brain is actually essential if we are to use these in a therapeutic context. So with that, I'd like to thank my collaborators and funding sources, and also mention that I would love to hear any questions that you may have, and you can email me here. Thanks.